that you did to prepare. It's not so easy to get a government person to travel. We have a lot of red tape since we invented it. So I'm here representing Kennedy Space Center. This is the logo of our group, Exploration, Research and Technology Program, the ISS program, the International Space Station program, and then the Veggie team. You'll see what Veggie is in a minute. And we're talking about food production for space exploration kind of generally, and then I'll go into more specifics on what we're doing right now on the space station. So if we're thinking about space food production, our goal is to produce safe, nutritious, and appealing food to supplement a stored diet. And that's our goal right now. So safety is critical. I don't want to be the one to get the astronauts sick, okay? I feel really, really bad. And we're growing these plants in an environment loaded with human-associated microorganisms. There's also the potential for greater levels of mutation in that environment, especially when exposed to galactic cosmic radiation. And so food safety has been really our main focus with the veggie research. We need to make sure that the produce is safe for the food we eat. It has to be nutritious, but when I say nutrition, I'm actually focusing on key nutrients that have been identified as either lacking or deficient in the stored diet, or things that don't store well. And if we're going to Mars, our diet is going to have to store for about 5 years. And right now we don't really have a good way to do that. And some nutrients will degrade over time in packaged food. Things like vitamin C actually degrades quite considerably. So they can enhance that in the diet, but we would also like to supplement that with fresh, bioavailable nutrients in the food. And then appeal is really important. I mean, if things don't taste good, you don't eat them. And the same is going to go for astronauts. They may play with it, but they may not eat it unless it tastes good. So we want to find produce that's really tasty. But we're right now just supplementing a stored diet. And the reason that we're doing this is there's no way to cook anything on space station. They don't even have a microwave. So we're really looking at produce that could just be picked and eaten fresh, maybe things like herbs that could be added to that packaged diet. But the longer we go and the farther we go, we might be able to add more components of the diet. So maybe in an intermediate time frame, we could grow things like sweet potatoes or white potatoes or beans if we had something like a microwave to cook them. Things that don't require a lot of processing equipment and that have a very high harvest index. Maybe when we have a permanent base on Mars, and we're there for a long period of time, we would grow almost the whole diet, right? If we can convince the astronauts that they can survive on a vegetarian diet or a completely vegan diet, we could produce all of the food for them. But that would take a lot of equipment, and it also takes a lot of labor. And as one of my colleagues likes to say, we're not going to Mars to be subsistence farmers. Right? We're going to Mars to explore Mars. So they're not going to have a lot of time to grow the food, to process it. You know, and the amount of equipment that you would need to take if you were doing crops like wheat or soybeans to process into flour, to make bread, tofu presses, all things like that, that's heavy. And it requires energy and other resources. So for now, we're thinking about this stepwise. So our key factors are really controlled environment production. Everything we do is going to be in some sort of controlled environment. We will not be using direct sunlight. There really aren't very good materials now for maybe a dome on the surface of Mars or on the surface of the moon. Most of our transparent materials break down very quickly under UVC radiation, and that will be out there once we escape the protected atmosphere of the Earth. They would be vulnerable to things like micrometeorite impacts as well. So we'll probably be growing in a protected environment. And we might use some sunlight, but it might be piped in with fiber optics, maybe parabolic mirrors. But it's also going to be lower. You know, the farther away from the sun we go, the less light there is. And if we have things like dust storms to contend with, that's even less light. So we'll probably be using a lot of electric lights. We will have high carbon dioxide. So on space station, right now, we're growing at around 3,000 parts per million CO2. And that's because you have 6 people living and working in a room. The CO2 in this room is going to just go up and up and up during our talk, right? And so these plants have to be able to deal with that. And that's a lot higher than what we have on Earth right now. we have to think about what are the common environments that multiple crops can share. We can't afford to have 50 different maximized or optimized environments for 50 different crops. We may at most have 2 or 3 different environments, and so we'll have to find crops or select crops that will all be happy within similar ranges of temperature, humidity, and things like that. And we also have to think a lot about scheduling those crops. You don't want 20 heads of lettuce one day a month, right? So you have to plan your scheduling for your crops so that you could have a continuous supply of food production. You know, with something like lettuce, you may always be planting and harvesting. With something like wheat, you don't want to harvest 1 head of wheat every day. So scheduling is going to be really critical. Some of the restrictions we have are power, mass, volume, and crew time. Okay? These are all costs. Costs to launch things and costs to operate things. So your system can't be very heavy. It can't be very energy intensive because that's a pretty precious resource when you're generating things from solar panels. Maybe we'll have a small nuclear generator on Mars, but still power will be limited. But crew time might actually be the most valuable one. It is the most valuable resource on space station right now. Even though we have 6 people living up there, there's really not that much time for all of the activities that they have to do. So we have to have procedures that are very minimal crew involvement. Sustainability is going to be pretty important. Right now we're not very sustainable with our production system. And we have to become more sustainable because we can't be flying up fertilizer, all the time. We need to be able to recycle inedible plant materials, recycle other wastes, find those, resources within those wastes to continue our growth. Our biotic stresses might be a factor. Hopefully not a huge factor. We're probably not going to take insects with us, hopefully, at least not intentionally. But, there are a lot of microorganisms in and around the crew, and those could be a problem. And I'll talk a little more about that at the end of this talk. But the abiotic stresses have been a real challenge, especially in spaceflight, where you have no gravity, because, there's no convection. Heat doesn't rise because there's no up. So getting movement of air, getting mixing of air and water is a real challenge. And we've had a lot of problems with fluids, which you'll see some more in the talk. So getting that environment correct is very critical for the plants. And that's really where it becomes kind of an engineering challenge to get all that to work. But I think there's a tremendous number of opportunities here. We can look at designing plants specifically for a spaceflight environment. This is kind of an experimental food system anyway. So we could have custom space plants that grow really well in this environment. They may not be very relevant for most terrestrial growing systems, but they may be perfect for us, whether those are from traditional breeding and selection from these super dwarf crops or from genetically modified crops. Automation, I think there's a lot of potential for automation, looking at robotics to try and take care of some of the tasks that the crew just wouldn't have time to do. And then what we call in situ resource utilization. So if we're on a planetary surface or even an asteroid or the moon, there are resources there. How could we use those in our crop production system? They've even found nitrates on Mars. So, there are resources that might be there. The Martian atmosphere, though very thin, has carbon dioxide. And then there's the regolith, which is the soil, and water. So all of these are really interesting opportunities that are speculative. And so when I think of the challenges, I kind of group them in space, microgravity challenges, surface systems like Mars or the moon, and then sort of crop challenges. And there's a lot of overlap in these areas. Some space challenges are like this lack of convection. How fluids behave. Water just forms a ball, and it will coat surfaces, which is a real challenge when you're trying to get both water and oxygen to plant roots. Dust and micrometeorites are going to be big challenges on the surface of Mars or on the moon. They'll interfere with a lot of systems. Dust gets everywhere, and it causes a lot of problems because it's not weathered. They don't have weather, so the dust is very abrasive and it's very invasive. For our crop challenges, some are the same that we're dealing with on Earth. How to get more productivity, more stress tolerance, but others may be a little more specific for our growing systems. How do we get very dwarf plants with a high edible to inedible biomass ratio? How do they grow in those high CO2 environments? So we have a lot of work to do, and there's definitely a lot of questions. But we pull in a lot of information from the general horticultural community, the general biology community, and then we can look at how that could apply to our research. So let's take a step back in time. In the 1950s, Topps put out a series of trading cards, and one of them was farming on the moon. And I really like this one. This was before we ever went to the moon. And they said farming on the moon will be mostly done indoors, under a large plastic dome, pumping carbon dioxide in, so okay. Sunlight will pour down on plants. Special types of plants might compensate for a lack of meat. And certain plants, like cactus, might actually grow outdoors on the moon. Well, we're probably not growing anything outdoors on the moon. But this was the front of that trading card. So it's probably not going to look much like this. You know, one thing you immediately notice is this is not a very high productivity system when you have your plants 10 feet apart. You probably wouldn't want to have to wear a gas mask in among your plants and a leotard, for that matter. You probably wouldn't want scary man-eating looking purple plants that don't really remind you of home. And you're probably not going to be in a big, open, transparent dome. Here's another rendition. This is a little bit later. This was from the 60s, Luna City. Again, you have a big transparent dome, which is pretty unlikely, but at least you're starting to get more densely packed plants. You've got some automation here. For a long time, we didn't think we would have an option to do fruit trees, but we've actually recently started testing on some dwarf plum varieties that only get about a foot tall. So maybe fruit trees will be in the work. There's a lot of time tied up in growing fruit trees, so they would have to be a very rapid growing plant. But at least you're in a shirt sleeve environment with the plant, so this is getting better. I like this one a lot better. Here you have your densely packed rows of plants. You're inside the habitat with the crew, where they're living and working and playing. And you have electric lighting here, feeding the plants. And you're in this very thick protective structure. So it'll protect from your micrometeorite impacts, your space radiation, and your dust. So I think this is actually starting to be kind of realistic for what a base might look like. This is on the moon. And here's a shot of one maybe on Mars. Again, you have your densely packed layers of plants under electric lights. Okay? So it will probably have to be pretty intensive agriculture like that when we're growing plants on an off-Earth base. But we're already doing this today on Earth. Here are a couple of shots from the Osaka Prefecture University vertical farming facility that my colleague Ray Wheeler took a few years ago. And you see the densely packed plants under electric light. So this is actually very relevant to a lot of the research that's going on in that environment. For a more near term, will probably be something a lot smaller, something like this, where you have your salad crops in a very small module, something that can be sent up on a rocket, and it might be connected to a small surface habitat. So this is starting to be pretty realistic, and people might go in and take care of the plants and then leave. Might have some gas exchange with the habitat. And we're doing this on Earth today too. This is a shot of the South Pole greenhouse at the U.S. Antarctic Station, and this is a pretty highly productive plant growth facility. University of Arizona helped design this facility and helped manage it in the early years, where they are producing salad crops for the crews as they overwinter in Antarctica. They have volunteer gardeners who go in and help out with it, and they have people that just go in because they want to be near the plants. They miss the smells and the sights and the flavors of being somewhere green. When you're surrounded by ice, that can be very important. So I think the psychological aspect of growing plants when you're living and working in these extreme environments might be even more important than the food that they're providing. They even have people who would go in here to sleep or to take dinner dates. So it's a really, it's a really mentally grounding thing for a lot of people. So now we're going to talk about the International Space Station, which is where I'm doing work right now. The ISS is our orbital which is nice. There are 6 people living and working up there. We hope to increase to 7 in the next couple of years. And we have laboratory space, we have habitation space, and then there's just a lot of storage space. And this is Veggie. So Veggie is a small plant growth chamber on the ISS. It's about yay big, okay? So it's not very big. We can fit 6 plants of something like lettuce in there. Veggie is our very simple chamber. So instead of completely controlling the environment, we're relying on the space station environmental control to provide temperature, relative humidity, and carbon dioxide. And we actually have, can't both see them here, but there are some vent screens down here. So air is drawn up through these screens, and there's a fan right in the center here, and then it comes out the top. So it's drawing a continuous supply of cabin air through the plant growth environment. Now, we have LED lights, and we have fans. Those are the only powered components for Veggie, and everything else is done passively. We wanted to see if we could do this with a very minimal power footprint. It's also collapsible, so it's a pretty low mass, low volume system. And what we grow things in are these little grow bags, essentially called plant pillows. They're filled with a substrate, which is baked ceramic, same stuff that's turfus that you probably use here, or profile. It's also used on sports fields and golf courses. And we put a controlled release polymer fertilizer in there. And then we plant our seeds in these little wicks, and everything is sent up dry. The astronauts unpack them, install them, and add water. And they also add water to a wicking reservoir that's on the bottom. So that was the original Veggie design, and it was launched to the space station in 2014. This was designed and built by Orbitech, a company in Madison, Wisconsin. And so for a lot of the early research, we tested plants in kind of an analog pillow system. So here are some crops that I tested. The red lettuce and the dwarf Chinese cabbage are actually crops that we're flying and testing in space. So for the Veggie 1, the very first hardware validation test, we selected the outrageous red romaine lettuce. We like the name, part of it, but it was also a very reliable germinating crop, grew rapidly, was a very palatable, highly acceptable, attractive crop, and it did have low microbial levels on the leaf surfaces. So if you think about it, it's pretty hard to wash your vegetables in space. So we want to keep crops that are pretty low with microbial content. It also has high levels of antioxidants because it's a red vegetable, and this could be a good countermeasure for space radiation for the crew. They have a diet rich in antioxidants. So we sent 2 sets of the lettuce plant pillows, 2 sets of 6, and 1 set of Profusion Zinnia pillows. And we selected those zinnias because, well, they're a lot of fun to grow. We wanted to send an inedible crop or something that wouldn't taste very good. They could technically eat it, but they didn't. But something that they could just grow and enjoy, because we weren't entirely sure what order they would be growing the crops and how long it would take us to get food safety approval. So I'll talk more about that in a minute. So Veggie was installed on the space station. This is astronaut Steve Swanson and Rick Mistracchio, and they're installing the Veggie in the Columbus laboratory. This is the European Laboratory of Space Station. And you can see what that environment kind of looks like. You know, it's very plastic and metal and very synthetic. So when you add purple light, it did change the environment. They actually would turn the lights off in the Columbus laboratory and just have the Veggie lights on, and they called it Club Columbus, so they treated it like a disco. But you can also imagine how having something green and growing in this environment might impact the crew. And one of the really neat aspects of Veggie that's different than a lot of previous plant growth hardware is that you can see right into the plant growth area. So our first set of pillows were installed, the water was added, the lights were turned on, the fan was turned on, and things began to grow. And then we had the crew do a couple of procedures. So after 3 days, we had them open up the wicks to help the plants emerge. These wicks sometimes stick together, and we didn't want to smother the seedlings. We put 2 seeds in each plant pillow, and after 7 days, we had them thin to 1 plant per plant pillow. And so far, things were going pretty well. 1 plant pillow failed to germinate. So water went in, but no plants came out. And we don't know why that happened, but just the luck of the draw. One thing you can see here, we did also have a temperature and humidity small portable data logger that was in there, so we could gather some data on the environment. So they thinned to one plant, but we again only had 5 growth. And now is where we start to get to the murder mystery phase of things. So after about 2 weeks, we had some major water stress. The plants were not getting enough water from the reservoir. So these plants in the back they're okay, they're still there, but these guys were really pretty much non-existent. So at this point, we were only getting photos once a week, and it makes it really hard. You'd get photos that were taken a couple days ago, and then you'd have to tell people how much water to add a few days in the future. So it was very logistically challenging. But we had the crew intervene and add water directly into the plant pillows because the reservoir was just failing to meet the needs of the plants and provide enough water. But we still lost these 2 seedlings. That one recovered, and those 2 were okay. So now we're down to 3. So it was sad, but it was, exciting at the same time. At least we had 3. We're 50%. That's better than none. But it was pretty stressful. But we had the crew just continue to add water directly to the plants, and it's not ideal. You're essentially adding lots of water so everything's wet, and then it slowly dries down, rather than giving a continuous moisture, which is what we would like. But it worked okay. And so after 33 days, the astronauts harvested. And so here's Steve Swanson harvesting the lettuce. It's a little bit pixelated. This ended up being a big deal on space stations. Normally those guys in the back and the one under the camera have other stuff to do, but they all gathered around and they watched the harvest and they took photos. And Steve was really proud, and he did a great job caring for these plants, followed instructions perfectly. So we didn't allow them to eat the lettuce for the first time because we wanted to get the food safety data. This was the first time veggie had been up there, and we needed to make sure that all those human-associated microorganisms didn't suddenly decide to colonize the leaves of our lettuce plant. So we had to bring the plant tissue back. He wrapped it in foil. You can see the foil sheet here, and it was brought back frozen. It was stuck in the freezer for several months on space station, and then we got it back. We can't guarantee that they didn't eat any of the lettuce, but we asked them not to, put it that way. It's very hard to make people do what you want when they're going 17,500 miles from the Earth. So we also got the data logger back, and here you can see some data on temperature and humidity. The red line is temperature, the blue line is humidity. We actually moved the data logger from the back left side of the chamber to the back right side where that one plant pillow failed to grow, wondering if there might be a hot spot. And it was actually a little bit warmer on that side on average, but not tremendously warmer. You can see here things are drying down. This is when our water stress was happening. But then once they started adding water, especially as the plant started growing, we get a little bit more humidity. So the bellows provide a little bit of humidity containment, but it's still pretty low, just 40%. So again, this is an environment for the astronauts, not for the plants, not optimized for the plants. With our samples, we were able to look at the fresh mass, identify the culturable microbes. This was really our food safety component. This was our priority. The plants, some water samples that we took, and the plant pillow components. We brought back 2 of the plant pillows and kind of dissected them. We identified the cultured microbes, but we've also done a total microbiome assessment of the plants. It's still kind of in work, actually, but we're doing the 16S sequencing as well as eukaryotes. And looked when we had enough tissue, we did anthocyanin, antioxidant, and phenolic analysis of the tissue, elemental analysis of about 13 different elements. And then this one was very cool. We did some X-ray tomography of the plants and pillows where you can actually image in a frozen plant pillow where the root distribution is. So that's something that we want to pursue further. I'm not going to go into all of that, but here you can see what the samples look like when they come back. Frozen lettuce, not the most exciting plant, but when we had enough tissue, we could do all of the analysis. But as you can see, our variability was huge, and our ends were very small. So it's a challenge. We did, however, get pretty good food safety data. specifically for E. coli, Staph aureus, and Salmonella, and we didn't find those at all in our space or ground produce. And in terms of NASA standards, there really aren't any standards for fresh produce yet, because no one had done this before. No one had grown in anything for eating yet. But we have similar standards for non-thermal stabilized food. So this would be like M&Ms or peanuts or things. And so we use those as kind of our general guide. And all flight plants were below the levels for aerobic plate counts. And all except one was slightly above for total yeast inputs. So we took all of these data and we presented them to the flight medicine board, the flight surgeons, the flight safety people. We have a lot of boards at NASA. And it took about 6 months to say, look, we think it's safe to eat. So they were approved to consume the produce with a precautionary wiping. We actually had some produce sanitizing wipes that we had sent up there to help clean the hardware, but we can use that to sanitize the produce. So with that as just a precaution, the crew were approved to grow and eat the second set of lettuce. So then about a year later, the second set of lettuce was started by Scott Kelly. He's the one-year astronaut that was up there, and Shell Lindgren maintained this crop. We modified the watering procedures. We basically added more water to start to try and establish our water column sooner, and we increased the photo frequency. So if there was water stress, we hoped to catch it way earlier. We did observe water stress again, but the astronauts intervened very quickly, and 5 plants grew. We had one, the pillow wouldn't allow water in at all, so that one ended up having some clogged lines. But 5 of them grew, and we allowed the crew to eat half the produce. So we still wanted some science samples back. So they harvested them live on TV, and we got our samples back last May. So here's a video of the harvest. This is Kjell Lindgren. Kjell's actually a medical doctor, and he really enjoyed the veggie experiment. He did a great job taking care of the plants. So basically we had them take off half the leaves and sanitize them, put them in between layers of sanitizing wipes, and then press it for about 30 seconds, and that was the sanitization. They tasted it with some olive oil and balsamic vinegar there. So it's all about the condiments, even on space stations. But this was the harvest, and it was pretty fun. But even he, who's an astronaut, tries to put something down, and it starts to float away. So it's a really different environment than you're used to working with. They have their feet in little straps on the floor so they don't float away when they're doing this. This is Kimiya Yue, the Japanese astronaut, who is running one of the cameras up there. And so they had a good time with it. And it was a historic moment, So Scott actually said, tastes like arugula. And then later he said, I don't really know what arugula tastes like. But they were here sanitizing the produce. These are the ProSan wipes. They put the leaves through. It's kind of a 2-person job for doing this, but they really enjoyed it. And they made some speeches, which was pretty exciting for us. So Scott talked about the logistical complexity of having people live and work in space and the long supply chain that you need to have, and how for Mars we have to become more self-sustainable with our food supply. And Kjell, who's a medical doctor, and he hadn't been there as long as Scott. He'd only just arrived partway through the experiment. But he talked about the benefits of fresh vegetables, the contributions of the plants to the mini ecosystem on the ISS, and really the psychological aspect of growing plants. So they were both really excited by this, and that gave us the opportunity to grow the third set of plants. So the third crop of zinnias were started in November 2015, and Kjell actually started them, and then Scott took over operations because, again, he was up there for almost the whole year, 340 days. So with these, we gave up on the whole wicking system. We just had them directly add water to the plants, and we decreased the photo frequency because people really wanted to conserve crew time, and so asking the astronauts to take photos every day or every other day was just kind of unreasonable. They have to upload them. So we decreased the photo frequency, which was a bit of a problem because we ended up giving the plants too much water. We also think the airflow in the chamber was somewhat obstructed or restricted at the time. So there ended up being quite a lot of free water in there, and we got some fungal development. Scott mitigated this, or he tried to mitigate this. He tried to cut off the damaged tissue and sanitize the plants, and it worked to an extent, but we still lost some plants. And then he said, you know, I'm the one who's here. I'm looking at these plants. I should really be deciding how much water to give them, not you people on the ground who don't know anything. And we said, yes, we totally agree with you. But there's actually these people in the middle. This is NASA, and we have levels of bureaucracy. And there are people in the middle who do all of the operations for payloads that are occurring on space station, and they wanted all the procedures written out, you know. So we would get a photo, and then we'd have to estimate how much water to add 5 days later, you know. So there was a big disconnect. So once Scott asked for this, we said, you know, we said, yes, we really want that, and we were able to both kind of convince these people to let him decide when to turn on the fans and how much water to add. So we started what's called autonomous gardening in December. And we gave him like a gardening guide. You know, if the plants are so big, you can just add, you know, this to this much water. I don't think he paid any attention, but he did a wonderful job taking care of the plants. He also asked if he could harvest them on Valentine's Day. And we said, sure, like it was Sunday, it was his rest day, but he wanted to do that on Valentine's Day. So he was just very gung-ho, and you know, we were thrilled. And we also got these plant samples in May. So what do they look like? Well, they didn't look like your garden variety zinnias. So one of the first things we ended up seeing was this guttation droplets on the hydrophones of the leaves, and some really dramatic leaf curling. And then it got even worse, and that's when we started to see the fungus. It was the 4 a.m. call on December 22nd when they said there's mold on the plants. And so then we had to drag ourselves out of bed and go in and communicate, and came up with a procedure to remove the damaged tissue and clean the plants. But you can see some of these plants are very stunted, and these are just really bizarre looking. These two plants got quite a lot of fungus, and ended up both being removed. This plant had some fungus, which was removed, but it recovered, and there's another plant in the back that did really well, and these two died. So now we're only down to 2 out of 6 plants. So again, we get this sort of murder mystery drama. But when they started flowering, it was a game changer. I mean, they were beautiful. And Scott took the plants all over the space station. He took it out, he took it for a walk, he took photos everywhere. They're called Profusion Zinnia for a reason. They just kept flowering and flowering and flowering. We ended up with 28 flowers on 2 plants by the time we harvested, and they were still producing 5. But they were just beautiful. We have a lot of action shots. So we took the plant in front of the Earth viewing cupola window. This is the 300 days in space with him and Misha, who's the 1-year cosmonaut, and they posed with our zinnia. And then they had flowers on the space dining table that night for dinner. So it just really shows that the psychological aspects of this were important to him. And you know, I think he also felt very empowered that he rescued the plants that us dolts on the ground were trying so hard to kill. So that was good too. Here's a shot. He asked if he could sacrifice a flower and take photos. He took about 60 photos of this flower, you know, in front of the window. And so we got a lot of data from this on long-term growth. The plants actually ended up growing for 90 days. So the lettuce had only grown for about a month. Microbial issues, obviously. We still had no human pathogens associated with these, so that was good. Plant pathogens, not so much. Flooding and airflow and how to maybe mitigate that. We wanted to see if flowers would develop in veggies, and they did. We weren't doing any pollination, but we wanted to see if seeds would form. And we did get some seeds forming. We haven't shown that they're viable yet. We actually have an intern right now working on trying to determine if we can get any viability out of these seeds. It's very hard to dry things down in space. And so when he harvested the flowers, we had him wrap them in a dry wipe and leave them in kind of an open bag, but there was still quite a lot of fungal growth in that environment because things just don't dry without gravity. It's very interesting. We looked at human factors, chemistry, microbiology, and plant pathogens. So it's actually been identified as a fusarium species, and we're now trying to figure out where that fusarium came from. Did we bring it, or was it already up there on the space station? But one of the other really cool things, when he harvested, he asked if he could make a bouquet. And we said, of course. So he made this beautiful bouquet, and he took photos of it in front of the window. And for me, this was a really poignant thing, because there's not a lot of opportunity to be creative and to create beauty, in space. They take some stunning photographs, but there's not a lot of other artistic, endeavors. And so here is the first on-orbit flower arrangement that he helps enable. So that was really cool. So our next steps, we are now doing what we call Veggie 3. There was no Veggie 2. So Veggie 3 launched this time last year, and then the plants just sat there. The seeds just sat there. But we did grow some lettuce starting in October. And this time we were using a cut-and-come-again repetitive harvest technique. So they actually had 4 harvests of the lettuce between October and December 28th. Some of those harvests were for food consumption and some for science. And the last harvest was split. We're looking at ways to pre-stage the plant pillows and send the seeds separately. So maybe the astronauts could just take up the seeds they want to grow in their pockets. But we're not sure if that would be an option. We think that could be really cool because we want to really make it a crew-involved activity. The Chinese cabbage, which was the one I showed you earlier, was actually selected because this is the top-rated leafy green of 8 that we tested in terms of growth and in terms of organoleptic acceptability. We actually grew a bunch of the produce and sent it to Johnson Space Center where they have a food lab, and they did taste testing, and everybody just loved it. It wasn't the most nutritious, but it was the most delicious. So that's why we sent that one. And since I made these slides, because we have to get them through an export control process before we present them, Peggy Whitson has started growing the Chinese cabbage on Space Station. So we actually have plants growing right now of this Scotia variety. But we've been doing this crop testing of leafy greens, dwarf peppers, and dwarf tomatoes, and these are all about this tall. We started that at Kennedy Space Center, and now we're working with Fairchild Tropical Botanic Gardens in Miami with their education and outreach program, the Fairchild Challenge. And they have 130 middle schools and high schools who are doing crop testing and providing us the data. So it's really allowed us to multiply the capabilities for finding new crops that can grow in space. They're also testing fertilizer, they're testing different harvest scenarios, so that's just been really valuable collaboration. And we have upcoming flights, Veggie 4 and Veggie 5, where we're actually going to have 2 veggie units side by side in Space Station, so we can look at different red to blue to green light ratios with the same crops and see the impacts of light on the Chinese cabbage and the dwarf tomato. So those will be in the next year or 2. We're sending the second veggie unit up this summer. So that will multiply our capabilities. Here's just a shot of Shane Kimbrough, who's the commander on Space Station right now. This was with the cut and come again harvest of the lettuce that's up there. And so I'll just wrap it up. So a lot of thanks. We have a large team of people that really enabled this to happen at Kennedy Space Center and Orbitech. The astronauts, of course, Steve Swanson and Rick Strachey, Scott Kelly and Shel Lindgren, and now Shane Kimbrough and Peggy Whitson. And the folks at Marshall, who make things a little challenging, but they're the ones who schedule and who do all the procedures, basically enable our science to work on Space Station. The Space Life and Physical Sciences program has been the main funder of this, but also the ISS program and the Human Research program is interested. And then this is just a teaser, because we are sending another much more capable plant growth chamber up very soon, called the Advanced Plant Habitat. So Veggie is very passive, very simple. The APH will have all the bells and whistles. It will control temperature, relative humidity, CO2, scrub volatile organics, have active nutrient delivery, measure soil, root zone oxygen level, have cameras, have many different wavelengths of light, and a lot of other sensors and control systems. So I encourage all of you, if you're interested, to look at NASA research announcements and consider, could we do our research in Veggie or APH on Space Station? Flying research. That's all I've got, and I'll take lots of questions.
Thanks, Dr. Massa, for the wonderful presentation. Any questions?
Yeah.
What was the CO2 level there? Was that the 3,000 or less?
It was an average for the first experiment average about 2,800. So You know, it's right around 3,000 for most of the experiments. They used to be higher. It used to be around 10,000 on space station, but the astronauts were having a lot of headaches and fuzzy thinking, and there were also a number of studies in submarines at the same time that the Navy was doing that showed if you had these very high CO2 levels, it caused a lot of, you know, kind of muddled thinking and slow response times. So they've actually really worked hard to try and get it lower. But this is with the systems running flat out to try and scrub CO2. So we keep saying if they grow more plants, they'll take care of all the problems. But yeah, we're actually getting some interesting high CO2 stresses with some of the crops that we test. You know, it might be okay at 1,500, but at 3,000, you start to get chlorosis or some other issues. So we're looking into that.
What is the non-gravity environment effect of these plants?
So it's really interesting that the plants don't really care so much about gravity. You know, in the absence of gravity, they just key in on their other secondary stimuli. So as long as we have blue light overhead, the shoots are phototroping towards the blue light, and they grow towards the blue light. The roots seem to be a little more disorganized, but they seem to grow towards the right proportions of water and oxygen. Obviously, we still have a long way to go with the watering system. We're in the process of developing a second watering system to try and solve some of those problems. But the roots seem to try and find a happy growing environment in terms of water and maybe nutrients. But yeah, they don't really, as long as you can get your environment good, they don't really care.
Yeah, at one point in time they had problems with ethylene accumulation. And so did you guys monitor that and try to mitigate it in this particular case?
We didn't actually. Since the space station has been built, they actually now actively scrub ethylene and other volatile organics. On the old Mir and on the shuttle, there were a number of problems with high ethylene, and they'd get like, you know, sterile wheat heads and things. But now they're keeping it below 50 parts per billion on space station. So we are not scrubbing. The Advanced Plant Habitat does scrub, specifically has a requirement to maintain it under 25 parts per billion. We didn't really have too much of an issue with that we've seen in veggie. Again, our sample sizes are pretty small.
Is there any genetic variation caused by the space condition?
That's a good question. We haven't tested that. We actually put a proposal in to look at transcriptomics of the lettuce. It didn't get funded, so we haven't tested that yet. I think that would be a really interesting question. There are a number of researchers who are doing experiments in veggie. I should say veggie is kind of a multi-purpose platform. So we have researchers working on Arabidopsis, and they can grow about 30 petri dishes in racks in veggie. We're going to soon have a flight using Brachypodium distachyon, the monocot model organism. Those will probably be growing in magenta boxes with kind of an oasis foam. The European Space Agency just launched, in January, they ran an experiment looking at lentil, mustard, and radish seed germination in a box specially designed that went in veggie. So I think those type of experiments are very feasible in this system, but we personally are just really more focused on the food crop.
So going back, you mentioned radish, and I've seen photos of you growing radishes on the pillows, which are really, really cool. But the radishes tend to grow on top of soil also fairly well.
Mm-hmm.
What challenges do you expect for growing other root vegetables that would not be so comfortable in an aerial environment in your pillows?
Right. I think we're going to have to design a special growing system for root crops, because the pillows really are designed to be very contained, contain all the particulate media. We don't want, you know, things floating into people's eyeballs. So we try and maintain that contained. But another big challenge, which is one of the reasons we haven't grown radish to eat up there, is there's a lot of ultrastructure that causes quite a lot of microbial growth on the radish. Even though they kind of look shiny, there's quite a lot of ultrastructure, and there's no good way to sanitize them yet. You know, a wipe is just not going to do it. So we have to figure out ways to do produce sanitization. If NASA doesn't relax the kind of standards, right now our standards are number of microorganisms, and anything in contact with water is going to have a very high number of microorganisms. We have been doing some testing with cold plasma sanitizing of produce to see if that could be an option. They were looking at it for cleaning like medical hardware and other things as well, but People have looked at it for produce. It seems to make a lot of things explode. So may not be so good, but we're still investigating that. But yeah, the way that fluids behave, you can't really do an aerosol because everything would just form a glob. And so it's a real challenge to figure out how to clean things. But yeah, so I think we need a whole new design probably for this.
You said that you didn't detect any pathogenic microbes, but just in general with the 16S and other assays, did you see significant changes in the microbial community in the flight grown versus ground control?
Definitely, yeah, we've definitely seen different community up there. So far, we haven't really seen anything that was surprising. So there have been a number of microbial assays on Space Station in the past, and all of the things that we've seen on the produce are typical Space Station microorganisms. But of course, when you do something like a 16S, you catch everything. And so we're still working through a lot of those data, and I am not I'm not the person to ask about those. So hopefully, we'll have a publication out about that at some point. But yeah, it's interesting, the ground community is very different. And so you can kind of think of this as kind of like an island biogeography situation. You have these new people show up and all their microorganisms are just distributed, but then there's some new equilibrium that's set, and you only get new people coming every 3 or 4 months. So when we try to send up the hardware, we try to send things very clean, but you can't get everything totally clean. So we'll see what's up there, but we do know we now have fusarium up there. So that's okay.
So given the current progress with producing food in the space, so how far in advance do you think we are away from have a permanent settlement on Moon or somewhere like outer continent?
Yeah, I wish I could tell you the answer to that. NASA's focus in the last 10 years or so has been on our mission to Mars. So we've actually stepped back at focusing on the Moon, and we're looking at trying to go to Mars, trying to go with a manned mission, and they don't even say to land, it might just be to orbit, which I think would be very disappointing, somewhere in the 2030s. So we're still about 15 to 20 years out. The problem is we are a federal government agency. And so when you get new administrations, they have new priorities and things change. And we're sort of waiting, for the other shoe to drop to see what may change. So we may end up going back to the Moon. We just don't really have a lot of control over that. But we're just trying to develop systems that could work in a number of different environments and trying to think strategically for the future about how we can always have some aspect of this food production. The life science research at NASA was really almost wiped out in around 2005. And that was when the administrator decided we didn't really need to be doing that, we needed to build a really giant rocket. And so basically they cut all the funding for the life science, pulled people's grants back and everything else. And so we're trying to rebuild and just become somewhat sustainable and show people, look, this is important. Yeah?
Would it be feasible or at all worthwhile to grow edible funguses in space?
I think definitely. Yeah, I think there's a lot of potential there. We are going to have inedible plant material. There's a lot of other resources up there, whether it's human waste or whatnot. So I think being able to grow edible fungi on some of the inedible plant waste would be tremendously worthwhile. Again, right now there's no cooking. Some of them you happen to eat raw. I think it would be very feasible because it would be a fairly low resource intensive thing, but we just need to find the right cocktail of things to put together.
Spoke a little bit about trying to recycle, not being able to send nitrogen fertilizer. Composting on ISS is something that NASA would be interested in.
Not sure about ISS right now. As you see, we're doing pretty small scale, but I think we definitely need to be doing that in the future. I mean, we're not going to send bags of fertilizer to Mars, right? So we're going to have to compost. Excuse me. I think there has been work in our group at Kennedy Space Center in the past on ways to, help stimulate this, promote it in sort of bioreactor situations. One of the big challenges is, trying to have a steady state supply. So until you get scaled up to the point where you're always having continuous production, hard to have a steady state supply of, inedible plant material. And, just throwing some in once in a while may not be ideal. So it's a matter of scale, I think. Yeah.
Would microgreens ever be a possibility?
That's a wonderful question. I actually have a scientist working with me now who's gotten totally in love with microgreens. And so we're really looking at them. You think about it and it's like, well, most of your stored energy is in the seed, so you'd have to take a lot of seeds with you. But the amount of light and other resources that they need are much lower than for crops where you're really using the photosynthetic potential. And the flavor and the nutrient levels are quite high. So I think there's a lot of potential. We have to convince the food folks. Most of the food people are like, what are those? We've never heard of them. The horticultural community knows about them because they're a very hot topic area, but the food people, they're a little slower. So we'll try to feed them salads whenever they come visit. Convince them.
Is anybody playing with the fusarium that came from Abiotic?
We do. We're working with a plant pathologist at University of Florida, Dr. Andy Sugar, and he's doing the Cox postulates testing to try and identify if it's, an opportunistic pathogen or if it's, an aggressive pathogen. And he's also looking at the different strains to try and figure out where it may be.
We'll take one last question.
You mentioned even looking at dwarf plums. What has been the thought on pollination for crops?
That's a really good question, and we're going to have to probably do it for our tomatoes. I think when you're working with a small enough system, the astronauts are the bees. They just will go in there and hand pollinate everything. The dwarf plums are an interesting challenge because right now the varieties that we've been testing, these were USDA variety developed. They actually require wild-type plum pollen to set fruit. So they're not ready for prime time yet, but we've been doing testing with them. It could, in theory, stay stabilized for a long period of time. But yeah, we're definitely going to want to have self-compatible crops, if at all possible. And, the astronauts are probably going to have to go in there and hand pollinate. They've done that in the past on Space Shuttle with things like the rapid cycling brassicas, and they had bee sticks, and they got to do the bees. So I think they like a little interaction as long as it's not too onerous.
Thank you so much, Dr. Massa. Thanks everyone for coming. And I also like to acknowledge the sponsors for this event, the Department of Horticulture and the Horticulture Grad Student Organization. Thank you so much again.